Look, I didn't really want to have to do this, but I'm going to have to address the elephant in the room. Yes, I'm talking about the coronavirus. That same virus that is forcing economic shutdown and worldwide death. Now in hindsight, we probably should have seen it coming. I mean, we keep talking about how pandemics are the greatest threat to humanity, but we never actually create any systems in place to ensure safety. But this video is not about the criticism of world governments and lack of preparedness. Instead, it's a project I've been working on, leveraging new medical treatments and technology to find a solution to these pandemics and disease. Most diseases directly affect your immune system and attack you from the inside. Most immune diseases can be eradicated because your immune system is either prepared for the disease because of vaccines or the disease comes in small quantities. Have your white blood cells to thank for your body's immune defense, more specifically, your body's T cells. T cells are a type of white blood cell that directly eradicates disease cells within the body. The way it does this is that it identifies dysfunctional cells and binds to the antigens on the surface of those disease cells. An antigen is just a toxin on the membrane of the cell. After binding with the antigen, it injects chemicals into the disease cell and triggers apoptosis, effectively killing the cell. Your T cells have done this all the time and have secured your body from multiple diseases. However, sometimes, some disease cells will have antigens that the T cells just can't bind to. And this becomes very problematic as the disease cells can easily multiply and spread throughout your body. This is especially common with cancer cells, where they trick your body into not having a proper immune response. Luckily, one medical procedure that is helping to solve this problem is CAR T-cell therapy. CAR T-cell therapy involves taking a patient's T cells and engineering them to properly fight the disease cell. The T cells are then multiplied in a lab and injected back into a patient's body. Specifically, the T cell's receptors are engineered to fight the disease cell. The T cell receptor, or TCR for short, is what binds to the antigens of the disease cell to kill it. The problem is that finding what parts of the TCR that will bind to the antigen, also called T-cell sequencing, takes a lot of research and money, which is the problem I tried to solve. And also, CAR T-cell therapy fails around 70% of the time because of this poor engineered cells. Essentially, using machine learning, can we predict the parts of the T-cell that will bind with a certain antigen? Okay, let's try to define this problem in more detail. The three most important parts of a TCR are the variable segment, joining segment, and CDR3 protein sequence. The variable segment and joining segment make up a large portion of the TCR, especially the connection to the actual T cell. The CDR3 or complementary determining region is a part of the variable segment and is what mostly dictates antigen specificity, or what the T cell can actually bind to. So essentially, we have to consider these three main parts of the TCR when actually engineering our T cell. And these can be our target variables. But what is our input? Looking at the antigens, there's one thing that mostly differentiates them and has a correlation to the parts of the T cell. The epitope part is the part that connects the antigen to the rest of the disease cell and is represented in the form of a protein sequence. Our problem becomes this. Given the epitope protein sequence of an antigen, can we predict the variable segment, joining segment, and the CDR3 protein sequence that will bind to the antigen? And the way I tackle this problem is I broke it up into two models. One that predicts the variable segment and joining segment, and one that predicts the CDR3 protein sequence. This is because the variable segment and joining segment can only belong to a handful of groups, which makes it a classification problem. And the CDR3 protein sequence can be an arbitrary non-fixed length of characters, which makes it more of a regression problem. And just a little context, a machine learning model is just a differentiable function that takes in an input and produces an output. It has parameters that it learns through a training process. This is where the word machine learning comes from. Now, one important thing to recognize is that the machine learning models only take in numerical values as input. We are trying to input the epitope protein sequence, which is represented by a non-fixed length of characters. To represent this as a numerical value, we can encode the characters by changing each character into a number. So for example, A becomes 1, B becomes 2, and you finally get the idea. So let's first look into how I did the classification problem and solved how do I find the J segment and V segment for the T cell. In terms of the actual model itself, you can see the architecture here. The first functionality that is incorporated into the model is the embedding layer, which takes our input and transfers it into a new vector of values. It does this to create links between our data and to figure out what is related to each other, making the machine learning process much easier. The other noticeable part are the dense layers, which are your standard machine learning layers. They have many parameters and propagate information into a last layer, which is 126 neurons, which represents the 126 classes that the variable segment can be. For the joining segment, there are 64 neurons in the last layer because there are 64 classes that this joining segment can be. Pretty simple, right? The last dense layer uses a softmax function that assigns a probability value for each of the neurons or classes in our last layer. The value is a number between 0 and 1. So if the neuron 43 has the highest probability, that means that the epitope sequence that has the 43rd class of the segment is probably the best for the TCR. This model has around 30 million parameters, and here's how the model was trained. Loss is just a way to represent how good a model is, and we want to optimize this to achieve the best loss as possible. The lower the loss, the better the model performs. So for both the variable segment and the joining segment model, the loss goes down pretty good, and it means that the models have pretty much learned the parameters. So let's also check out how accurate the model was. And it was actually quite poor, with an accuracy of only 20%. However, this isn't the full story, as there are a multitude of segments that will work for a certain TCR. So the real accuracy is actually around 80%. So let's actually try to predict the variable segment and joining segment for the yellow fever virus. This is the epitope protein sequence, and when we put it through our model, we get these two segments. Now the model for predicting the CDR3 protein sequence is a little different, since it's not a classification problem. Instead, I use the sequence-to-sequence model, which does what it sounds like. It takes in a sequence of a non-fixed length and produces a sequence of a non-fixed length. The way a sequence-to-sequence model works is that it uses an encoder-decoder architecture. The encoder does one specific thing. It takes in the input and extracts the most important features from the input. It does this by using an LSTM, which can keep track of a long-term memory state, short-term memory state, and cell state of the specific data input. Extracting these pieces from the input data allows the decoder to construct it into an output. The decoder does this by using a combination of dense layers and LSTMs, but instead, the output of the LSTM is not the actual cell states, but instead the actual output of the LSTM, which is what we want at the end of the day. Encoder and decoder models work so well because they can correlate input and outputs and produce a non-fixed length of output as well, which is exactly what we were looking for, and this is exactly what we got. The input for this model is represented in the same way as the dense model, but instead, the input is also one-hot encoded. A one-hot encoded vector is an array of zeros, with one number in the array as a 1. The position of the 1 in the array represents the class of that one-hot vector. Since there's 26 letters that a letter could be, the length of the one-hot vector we want for our problem is 26. The output this time, since it's not class-based, is an encoded array. 1 is A, 2 is B, and so on. The way it produces a non-fixed length is that it uses an end token as part of its output. It can output an end token which tells us that the sequence is done being predicted. This end token is what allows for a dynamic output to happen, which is what we need for a CDR3 output sequence. So here's the model architecture I was talking about. You can see the encoder states are taken out and fed into a decoder. When training this model, the output is used as the input for the model, another property of sequence-to-sequence models. And here's how the training went for the loss. The loss went down very well and got down to a very good loss. The accuracy is the same story, as it rose to 80% for the training data, which means that it can predict the CDR3 sequence to an 80% accuracy, which is very fantastic. So let's demo this model using the yellow fever virus. So we insert the epitope protein sequence and we wait for it to predict it. And we get a CDR3 protein sequence of CSGTGTGELFF. This is actually the CDR3 protein sequence for this actual virus. So this model works indefinitely, and it works really well. So what does this all mean? It means that we are one step closer to adjusting that failure rate of 70% for CAR T-cell therapy all the way down to 0%. Machine learning has the ability to allow for more precise engineered parts of TCRs that will help get the failure rate down hopefully to 0% one day. And the thing is, these models definitely prove that we can get there with the help of everyone. AI and machine learning is already revolutionizing new medical procedures, but it is applicable to much more. AI will indefinitely make a large impact that will save billions of people, and this is just one of the examples. Thank you.